In the nucleus of the cells, you can find the DNA. Every day, the DNA of each cell is damaged thousands of times. If only one strand of the DNA is damaged, this can be easily repaired. PARP proteins bind to the damage and attract other proteins that repair the damage. When damaged DNA duplicates before the damage is repaired, a double-strand break can arise. In a healthy cell, this more severe damage can be repaired with the help of homologous recombination. BRCA1 protein binds the loose ends of the DNA and starts to unravel the DNA strands. Using the undamaged DNA strand as a template, the damage is then repaired. Finally, the two strands are separated, leaving both DNA strands error-free.